Fraser is going to talk to us about larval assessment threats and threats and opportunities. All yours, Fraser.
Thanks, John. Hear me okay in the back there? Okay, looking good. Thanks, Lisa. You set me up pretty nicely wherever you got to. I can feel for some of those dams failing with some aging infrastructure some days after 50 years. It was like, yeah, I'm there. Anyway, yeah, I'm going to talk about threats and opportunities with larval assessment. I acknowledge my co-authors here, Nick Johnson, Lexi Sumner, Becca Phillips, and Aaron Jubar. So an overview of my talk here. First off, what is larval assessment? Why is it important? And then I'll jump right into some threats in terms of the challenges we face in terms of what streams to treat. Climate change, we've heard about this before, and stream access too is important for us. And I'll finish off with some opportunities, particularly talking about environmental DNA and some knowledge gaps in terms of population dynamics. So what is larval assessment? We've heard a bit about this already. Don talked about it this morning. If we want to boil it down as much as we can, basically we find the sea lamprey. We have a couple different tools we use to do that, but what we're looking for is the distribution of the lamprey not only within the streams but across streams, so looking for new producers. And we also look at the age and size composition of the larval populations. These are important in terms of selecting the streams that we want to treat the subsequent year. I'll talk more about that in a bit. So the different tools that we use, we have three sort of main tools in our toolbox. The first of which is the backpack electrofisher. The top picture on the right there shows a couple of our guys at work doing some backpack electrofishing in a shallow stream. The lamprey, as we've heard, live in the sediment on the stream bottom. For deeper or more turbid streams, we use the application of granular balicide. I may call it GB at some point during this talk. And the way we sample with this tool is applying the balicide into small sample areas. The lampricide is coated on the sand granules. The sand sinks to the bottom. If the chemical lampricide dissolves off of the sand granule, lampreys are exposed to it, irritated, swim to the top. We sample them that way from boats. Our third tool, I don't have a picture of it, the deepwater electrofisher. We use this a lot on the St. Mary's River. It allows us some good quantification methods in some of the bigger systems. So why is larval assessment important? It basically drives the control program. Like I said before, we set out to find the streams that are infested, and those that are infested, we can't treat them all every year. We just don't have the resources to do that. And so what we need to do is prioritize those streams that give us the best bang for our buck. The other thing that we do also in terms, we generate the treatment list, but also we need to find the best application point for the lampricide within those streams that we're going to be treating. So that's a key part of the larval assessment game that we undertake. So we don't want to overtreat a stream, obviously. We don't want to expose more of the stream than we need to the lampricide, but we don't want to miss larval populations. So we need to find that sweet spot for those lampricide applications. Also, after a treatment, larval assessment teams go in and determine if lampricides are effective. We do generally have really good success with these, with most of our treatments, but we need to assess that and make sure that we continue to be effective. So jumping right into the threats that we face from larval assessment, one of the kind of different things that came up when I was thinking about this has to do with, we're seeing a bit more of a mismatch lately than we used to be in terms of the populations of larvae and adult sea lamprey abundance. There seems to be a diminishing returns, I'll put it, for some of our larval surveys. It's a good problem to have, right? We can't find lamprey. To the point where we're actually raising lamprey for experimentation. I'll talk about that in a little bit. But so there's certainly sea lamprey out there, and we're able to generate lists of streams to treat. But it's not like the bad old days, I'll call them, where we could not that long ago it seems like go out and find plenty of large larvae. And on the adult side of things, we're not seeing a corresponding drop. We have a pretty good control, we feel, on the adult abundance, But like I said, we're not seeing that corresponding drop in terms of what we're seeing in the larvae in the streams. And the potential reason for that, of course, is you might be thinking, well, maybe there's the same number of larvae, but they're just surviving better. And that could be the case, which leads me to some other threats that are linked to that, one of which we've heard about already, climate change. Some of the potential impacts that we're faced from the larval side of things has to do with increased growth rate and, like I said, survival with potentially warming streams and lakes. We could also see increase in the number of infested streams that were previously too cold for larval survival. So particularly in Lake Superior, there are streams that we think are not infested by sea lamprey just because they're too cold for the larvae to survive. Obviously, with a warming climate, this could change and we could see an increase in the number of infested streams. Also, within stream distributions, if some of the streams have cold headwater areas that are not currently suitable for sea lamprey larvae, with a warming climate, that could change and we could have to expend more of our resources assessing and controlling those areas. Lisa talked a lot about this one in terms of increased flooding and associated high water that goes along with those. That can make our work difficult. Maybe our results become unreliable. And also, of course, with the higher water level, the higher water events, we could have increased barrier breaches that Lisa talked about, and this would result in the associated range expansion of the larva. So stream access is really important for the whole program. Larval assessment not excluded. We need to be able to get to these streams to be able to assess them. So we need to be able to continue to get concurrence to access the streams. Species at risk seems to be becoming more and more of a challenge in terms of increasing concern for some of the species at risk. There have been impacts, especially in terms of the application of granular bailicide, which if there's limitations on what we can do with that sampling approach, we might be forced into using electrofishing more often, which is not effective in some of those environments. We've heard a lot about social license already. I'm going to talk about it some more. Just sort of generally defining it as a diminishing public recognition of the importance of larval assessment, sea lamprey control in general, but for this talk, specifically for larval assessment, and potentially increasing sensitivity to the application of granular bailicide. Also linked to the potential loss of social license is the turnover of partner agencies. Right now we have pretty darn good relationships with most of our partners, and with high turnover or changes in our social license, this could be at risk in the future. So now getting into some opportunities, how do we make what we have, we think is pretty good sea lamprey control right now, how do we make it even better? So quickly here, we've already talked about trying to increase the cross-training that we have for our staff. We have good knowledge in our different crews, and we have 3 different offices around the basin, so we've talked about trying to enhance the cross-training and spread that good knowledge around. We have tremendous amount of data going back 50 or 60 years, and there is possibility to better leverage those data sets. Also, further engagement with researchers to address some of the knowledge gaps. I'll talk about this more in a bit here. And lastly on this slide, the adoption of new and emerging technology, in particular environmental DNA or eDNA. We've already started down this road. Things are looking good so far. We've had some pilot studies done, and we've had the establishment of genetic markers now. So in these pilot studies, we've run the eDNA alongside with our standard assessment methods, and they're comparing really well. So this is good news for us. There's certainly more work ahead of us in terms of refining the sample protocol and the processing of the samples. But there is what we consider to be some huge potential to save considerable time and effort from the larval assessment side of things, particularly with what we call distribution and detection surveys. The distribution surveys are those where we're trying to find that sweet spot for the application point. So with some water sampling, we can maybe refine that a lot better and save some resources. and detection surveys where we're looking in what have historically been non-producing streams. We can sample those potentially quite quickly with eDNA. Looking to the future, this is sort of pie in the sky right now, but if we could quantify larval populations using eDNA, that would be super nice too. Not in our toolbox right now, but certainly an opportunity. I'm going to get into some more science-based slides here in terms of the opportunities and working with researchers. So the Sea Lamprey Aquaculture Program, run by John Hume out of Hammond Bay, they've been doing some really cool stuff. They've been able to complete the life cycle in the lab from spawning lamprey up through to metamorphosis. And with the lamprey that they generated, they've been able to already increase our understanding of larval growth, survival, metamorphosis. And an example of this in this next slide here, this is some of the results that they presented last year. And this is regarding the question of does density influence growth and survival. So the picture here are lamprey brothers and sisters, you could say. And the number per tank is those numbers in the red tape. So as we go down the image, we've got some numbers here that indicate the average length of those lamprey at different densities. So the low density at 15 per tank, you can see that they're pretty big larvae after 9 months, surprisingly big. And then the survival, as we go into more and more dense rearing scenarios, the lamprey obviously gets smaller, and their survival actually is surprisingly high. But in answer to the question, does density influence growth and survival? It certainly does. And the cool part of this is that all these lamprey, like I said, they're brothers. They're all spawned, and sisters. They're all spawned the same day from the same parent. So we can eliminate the genetic portion of that question and know that it's completely environmental. Another question that they've already looked at is, will lampreys grow faster in warmer streams? This links back to the climate change, obviously. This is 3 different temperature scenarios and a couple different diets that they were put on in the lab. The question, will lamprey grow faster? You can see that the 3 different temperatures, 8, 15, and 22. The warmer it is, the bigger they're getting. The 2 different diets didn't seem to make as much as a difference. It was clearly the temperature. They also found that there was higher variability in the growth at the higher temperature. So they've opened some real, some large doors in terms of the questions that we can answer now with the SLAP program. This is getting more into the SUPCON project. This is some work of Nick Johnson, and this is a somewhat confusing slide when you first look at it, but I'll do my best to explain it here. And these are 2 different streams and parentage or pedigree assessment of the wild larval population. So if we start with the picture on the diagram on the left, what this indicates is the parenthood of the offspring, which you see in the middle. So the take-home message from this diagram is that there are many spawners in the lower Black Mallard River over many years. And we can contrast this to the Okiyok River. Those 2 circles on the bottom left of that diagram indicate that there were 2 prolific adults that contributed to the overwhelming majority of the offspring in that stream. So the opportunities here, of course, are using this approach, the SUPCON approach. They can really get at the reproductive contributions of individuals, and also we can dive really deeply into larval growth and survival questions. So like I said, this is some of the output from the SUPCON project. So in summary here then, larval assessment is a pretty important part of sea lamprey control. It drives the treatment program essentially. We need to acknowledge our threats and take advantage of the many opportunities that we're starting to get into already. Thanks.